Hello friends, myself Professor Khemchand Surana. Today we will discuss about the synthetic antibacterial agents fluoroquinolones. In last lecture we have seen about the quinolones synthetic antibacterial agents. Now today we will discuss about the fluoroquinolones. The fluoroquinolones are a relative new group of antibiotics. So in quinolones, we have seen that the second generation of quinolones in the classification we have seen from the second generation of the quinolones, they were relatively called as the fluoroquinolones. So these are what the advances into the fluoroquinolones, which are a relatively new group of antibiotics. They were first introduced in 1986. So that in 1986, the first time these fluoroquinolones were introduced, but they are really modified quinolones. So the whatever the quinolones were there on that basic moiety, the modification over there, which is what the class of antibiotics whose accidental discovery occurred in early 1960. So these quinolones were accidentally discovered as a antibacterial agents. So the accidental discovery, so we can call these quinolones or fluoroquinolones as a serendipities. So here the basic structure of the fluoroquinolone is given. In that, into the red color there is a fluorine and their substitution R. In the classification of fluoroquinolone, we have seen there are 4 generations of fluoroquinolones. The first generation, where is what the older or oldest class of the quinolones. From the second generation of the quinolones, these quinolones were called as a fluoroquinolones. For example, from the second generation, there is a ciprofloxacin. There is a fluorine group is substituted, so we can call this ciprofloxacin is a fluoroquinolone from second generation. From third generation, there is a levofloxacin. Again, there is fluorine group is substituted over there. So these are what the examples of fluoroquinolone. So in examination, they were asked about the classification of fluoroquinolone. So you just have to remember the examples from the quinolone which were substituted with fluorine, and you have to write the same examples into the fluoroquinolone class. Now we will directly move to the mechanism of action of these fluoroquinolones, which is similar to the mechanism of action of quinolones. Also, these were block the topoisomerase as well as DNA kyrase enzyme. So it blocks the bacterial DNA synthesis. So whatever the DNA synthesis process is there, that will block by the fluoroquinolones. The blocking of this DNA synthesis by the inhibition of bacterial topoisomerase second, which were also called DNA kyrase enzyme, and also the inhibition of topoisomerase 4. The inhibition of ATP dependent DNA kyrase. So they were blocked basically. The whatever ATP dependent DNA kyrase is there, that will inhibited by these, which will Nike the double stranded DNA. Nike means the cutting off the double stranded DNA. So they were cuts the double stranded DNA and introduced into the negative supercoil and then reseals the Nike DNA. So whatever the after the nicking of double stranded DNA, these fluoroquinolones were introduced into the negative supercoils and then again reseal these Nike ends. So this is required to prevent the excessive positive supercoiling of DNA strand when they were separate to permit replication and transcription process. So this is what the prevention during the process of replication and transcription. The inhibition of DNA kyrase also prevents the relaxation of positively supercoiled DNA. We have seen in the last slide they were attached to the negative supercoil, so they were also prevent the relaxation of positively supercoiled DNA. the inhibition of DNA nicking closing enzyme responsible for DNA elongation. If we inhibit this nicking or closing enzyme, which will what responsible for DNA elongation, so which leads to what the break into the double stranded DNA. Inhibition of topoisomerase 4 interferes with the separation of replicated chromosomal DNA into the respective daughter cells during cell division. So also we have seen that these fluoroquinolones were inhibit the topoisomerase force, which will interfere with the separation of replicated chromosomal DNA into respective daughter cells during the cell division processes. The critical imbalance in the cellular metabolism resulting from the inhibition of enzyme participate in the sequence of cellular events which may lead to premature cell division, delay cell division, total failure of cell division leading to the lysis of cell. So this is what the regarding the cellular metabolism, which leads toward the inhibition of the enzyme which will participate into the cellular events, and this leads to these 3 points which are we have seen. The first is what the premature cell division, delayed cell division, and total failure of cell division, which will lead to the lysis of cell. If the lysis of cell will be occur, there is what inhibition of DNA synthesis occurs. Now this is what the pictorial diagram of mechanism of action of fluoroquinolones. Again there is what the double stranded DNA is there. There is what topoisomerase enzyme, DNA gyrase enzyme is given here. The fluoroquinolones where comes and binds to the two nuclear enzymes. Two nuclear enzyme means what? These topoisomerase 4 and DNA gyrase enzyme, which will inhibit the DNA replication process. So this is what the simple pictorial diagram is showing the mechanism of action of fluoroquinolone. So now again this is the flow chart given for the better understanding of mechanism of action. So fluoroquinolone is there, which will bind to the A subunit. In quinolone case we have seen there are two A subunit and two B subunit we have seen. So here is what the binds to A subunit of DNA gyrase means what topoisomerase second site, which will prevent the binding of substrate to the active site of DNA gyrase. So we have seen in this pictorial diagram there are two enzymes topoisomerase 4 and DNA gyrase. They are having a binding site. So when these fluoroquinolones were bind to the A subunit of DNA gyrase enzyme, that will prevent the binding of substrate to active site of DNA gyrase. If this binding will occur, there is absence. If this binding will prevent it, the absence of formation of enzyme substrate complex is if this complex will absent over there, there is the blockade of unwinding of double stranded DNA into single stranded structure. So if this blockade of unwinding of double stranded DNA occurs, there is directly prevention of synthesis of mRNA. If this mRNA synthesis prevented, there is inhibition of bacterial protein synthesis. If this bacterial protein synthesis is inhibited, means for the fluoroquinolone is showing the antibacterial activity. So this is what the mechanism of action of fluoroquinolones. Hope all of you get it. So now the next point we will go through that the resistance of these fluoroquinolones. So these are basic 2 points which are showing the resistance. The resistance appears to be the result of. So these two results were occur during the resistance. The first point is that the alteration in the quinolone enzymatic target. So basically what the whatever the enzymatic target is there, there is alteration. If the alteration will occur, decrease outer membrane permeability. If the outer membrane permeability will decreases, or the development of influx mechanism, so this influx mechanism will develop. the one or more point mutation in the quinolone binding region of the target enzyme. So again, the regarding mutation in the quinolone binding region of the target enzyme, or from a change in the permeability of the organism. So these are what the 2 points we will showing the resistance of your fluoroquinolones. The next point is that the pharmacokinetics. So again, the pharmacokinetics, the first thing comes into the mind, that is what the absorption. So these are well absorbed by the oral administration, and having 80 to 95% of oral absorption is impaired by divalent cation. So oral administration is there, and near about 80 to 95% of oral absorption is there. So distribution, so these were widely distributed into the body fluids and tissues, but limited CSF penetration. So widely distributed but having a limited CSF penetration. It can pass the placenta reaching to the fetus. So if there is basically these drugs were past the placenta and they will reach to the fetus. So half life near about is 3 to 8 hours into the serum. Next point is what the elimination, 30 to 50% from the urine by tubular secretion or glomerular filtration, and some amount into the bile and feces. So maximum is from what the urine excretion is there, and after that the bile and feces is there. So next point is came, that is what the uses of fluoroquinolones basically. So you can explain same uses for quinolones as well as fluoroquinolones. The first use, that is what the respiratory tract infection. So we will go through with the systems. The first is what your respiratory tract infection. So first is emphysema. The emphysema means what basically? The collection of pus into the cavity of the bodies, especially in the pleural cavity. The pleural cavity location is area between the lungs and the inner surface of the chest wall. So in case of emphysema, these drugs were used. Second is pneumonia, the infection of lungs that caused by bacteria, viruses, fungi and parasites. The pneumonia is what? The infection of lungs. Next is lung abscess, bacterial infection that occurs into the lungs tissue. So whatever the lungs tissue is there, in that the bacterial infection, that is called as the lung abscess, which will cause the tissue to die and pus to collect into that space. So these are three basic infections where these fluoroquinolones were used in case of respiratory tract infection. Next is meningitis. Meningitis means for the inflammation of lining of brain and spinal cord. The third use is regarding urinary tract infection. So in that, the first is pyelonephritis, a type of urinary tract infection. So this is what the pyelonephritis. This is the urinary tract infection that affects one or both kidneys, which will cause by bacteria, mainly Escherichia coli, or simply you can call E. coli or virus infection. So it causes the kidney to swell and may permanently damage them. Next is epididymitis. Inflammation of epididymitis, a tube near to the testicles that stores and carries sperm in case of males. Next, prostatitis, inflammation of prostate gland due to urine infection in the men. Next is cystitis, which is what the inflammation of bladder usually caused by urine infection into the woman. So these are regarding the urinary tract infection. Next is gastrointestinal tract infection. In that, first, enteric fever. Enteric fever means what? The potential fetal multisystemic illness that will cause primarily by Salmonella species. Second is bacterial diarrhea, which will cause by the Campylobacter, Salmonella, Shigella organisms. Again, in case of skin and soft tissue infections, these are used the infected ulcers and infected burns. So infected ulcers basically the shallow wounds that develops on the skin, and infected burns basically the red color and warm to the touch due to the infection. So these are what the uses. Again, the some basic. The next is what the gonorrhea. Gonorrhea is what the venereal disease involving in the inflammatory discharge from urethra or vagina. So this is what in case of gonorrhea is also gonorrhoid, the bacterial infection that causes open sore on or around the genitals of men and women. So next, tuberculosis infection, bacterial disease characterized by the growth of nodules in the tissues, especially into the lungs. Conjunctivitis, inflammation of outermost layer of the white part of the eye and the inner surface of the eyelid. Thank you. Happy learning. Wish you all the best. If you have any query, you can ask me.